So that means it surrounds the entire cylinder. Are we good on that?
Yeah.
So that means calcium is in storage outside of the myofibril, not inside. That's going to be important coming up. Next, see the yellow tube?
Yeah.
These aren't blue and yellow in real life, okay? But this yellow one is trying to point out that there is a tube that runs roughly the length of a sarcomere. And these tubes, how would you describe them, how they run?
Perpendicular.
Perpendicular to the
Muscle fibers.
length of the fiber itself. And so these tubes are called T-tubules. T, which is a perpendicular. Okay? So these tubes are perpendicular to the length of the fibers. It's a T-tubule. That will come back in a few minutes as well. All right, are we good on the structure?
Mm-hmm.
Now I'm moving. Next.
Move.
All right, now I hate this drawing, but it's a necessary evil. Sarcomeres are not two-dimensional as this diagram depicts, but I kind of need to show it as two-dimensional because if it shows up on your DCA, I want you to be prepared. So what's this?
Sarcomere.
Sarcomere, but it also is the A band. Okay, so it's not the sarcomere. Dang it, you made me misspeak.
Sorry.
This is the what? What protein is this? That's the myosin. The length of the myosin filament is the A band. It's officially the anisotropic band, but within the sarcomere we have some specific areas. The one that runs the entire length of myosin is A band. Where there's only actin, it's isotropic or I band. I will always call it I or A. Those are the two most important bands. How do muscles work? By getting shorter. New stuff. Ready? Hanging off the myosin. See these little things?
Mm-hmm.
They're called myosin heads. Myosin heads. They will be important today to understand what the heck is going on. Myosin heads, I want you to think of like Vikings ships. You know how they all row together?
Uh-huh.
Okay, guess what these are going to do? I'm going to sum up muscle contraction simplistically right now, and then I'm going to show you more detail, okay? Guess what these myosin heads do?
Flip over.
First of all, they must attach to an actin. And then they go, what would that do?
Move
But it would
closer.
pull these actins where? This way. Guess what happens to these myosin heads? They attach and they pull the actins this way. And so the whole thing gets shorter. What do you think would happen to the A band itself?
Shorter.
It stays the same. What happens to the I band?
That one gets pulled
It
closer
disappears.
to.
The I band will disappear completely, and the A band never changes because the myosin itself never changes. So we have muscle contraction where the length of the actin and the length of the myosin never changes. Neither one actually changes length. One gets pulled over the other. And I'm going to demonstrate it here in just a little bit. Let's go to the next one. Click the link. Quick review. You're going to have to click the play button down here. Okay. So go ahead and click it. It's going to light up some things, okay? Click it again. All right, what's this that it just lit up? Myofibril. What is this whole thing then that I'm touching?
A fiber.
Muscle fiber. But this is only showing 5 myofibrils. Is that accurate? No, there's a lot more, right?
Yep.
What would be in here?
Actin.
Actin and myosin. Myosin is thick, actin is thin. What's from here to here?
A
Sarcomere. So that means sarcomere is arranged end to end to end, right?
Yeah.
Okay, what's the yellow stuff?
The sarcomere.
Sarcoplasmic reticulum. And what's in it? Calcium. What's the blue tubes? T-tubules because they run perpendicular to the length of the fiber itself. All right, I'm throwing a lot at you, but that's like second or third time I've said it, right? I'm trying to repeat a bunch of stuff as we go too. Get out of this and go back to the present. And, whoa, yeah, just do that. And then next. Okay, stepping it up. We're about 20% of the way through new material. And tough stuff. We can do it. Go team.
Alright.
You could have divided this into 2 days.
Neuromuscular junction. So that means it's where a neuron meets a muscle. This should be review. This is a neuron that connects to, what do you think this is down here? A muscle. Hence the name neuromuscular junction. Alright, muscles, skeletal muscles are what? How did I just describe it 2 minutes ago? Voluntary. Therefore, where does it start? Where do we start the contraction? Like if I want to contract biceps brachii,
Brain.
where did it start? In my brain. In particular where?
Motor cortex.
Motor cortex, which is part of what lobe?
Frontal.
Frontal. Motor in the front, remember? Frontal lobe. What's the name of the gyrus?
Central. Pre-central.
Pre-central
Central.
gyrus. Holy cow, he's asking us Unit 2 stuff? Yes. Yes, I am. So, what kind of neuron is this? That means it starts up on my motor cortex, goes down my spinal cord, and then out what kind of a neuron? Motor. Every once in a while it's good to read. Motor neuron. Why isn't it sensory? Because it's not sending information to the brain. This is a motor neuron. What's another name for the motor neuron? Efferent. Efferent. Okay, so action potential is coming down from the brain, from the spinal cord, and eventually it's going to get out to this motor neuron. A motor neuron that will end, like if I want to make my biceps contract, by the way, everything I'm teaching you takes place in like 1/10 of a second. If I want to make this biceps contract, I have to send that message, action potential will come down a motor neuron, and it will end right here. What happens right here is magic. Let's learn it. Here comes our action potential. Ooh, good thing they're smiling because it what? Acts as a what? Insulator, which does what to the action potential? Speeds it up. And why these spaces? What are these called?
Oh,
I
darn.
forget everything.
Nodes of
Ranvier, unmodulated areas that boost the charge. So once our action potential gets all the way down to here, I taught this before too, so this should be reviewed. What is this right here? It's a what? Voltage-gated calcium channel. Good to read. Voltage-gated calcium channel. What happens when the action potential hits it? It opens up this, whoop, and what comes in?
Calcium.
By what process?
Diffusion.
By diffusion. All we had to do was whoop, open the gate. Calcium comes in, attaches to what?
Vesicles.
Vesicles. When calcium attaches to a vesicle, it causes the vesicle to, look at the arrow, fuse with the cell membrane and release its contents. What's inside?
Calcium.
Did you need me? Right now? So what happens? What gets released?
Calcium.
No,
Attach.
calcium attaches and it releases a neurotransmitter. Remember, there's neurotransmitters in here.
Sure.
This, ring a bell?
Yeah.
Neurotransmitters get released. In this case, muscles have a very specific neurotransmitter. It's written here, it's on your paper. Acetylcholine is how you say it. Acetylcholine. I'll abbreviate it ACH in most cases. So, back it up. Action potential propagates down. What happens when it hits voltage-gated calcium channels? Calcium comes in because it opens up the gate. How does calcium enter? By what process?
Diffusion.
Because it goes from a
High to low.
high to low. Calcium comes in, attaches to a vesicle, causing the release of neurotransmitter. In this case, it's called acetylcholine. Where does it go? Acetylcholine comes across and attaches to?
Sodium. Potassium.
Stop.
Darn.
What is this? It's the muscle cell membrane, which was named? Starts with an S. Sarco. Oh! No! Sarcolemma. Sarcolemma. And so this whole thing is the cell membrane of the muscle called what?
Sarcolemma.
There we go. Now what? Acetylcholine comes in and attaches to receptor proteins on the
Sodium.
sarcolemma. So these are receptor proteins. What happens when acetylcholine attaches? Looky, looky. Look. Sodium comes
In.
Therefore, it's showing you sodium entering. Why would sodium come in? Because it's going to go from what? Therefore, it must be in a high concentration where? Outside. Therefore, the charge of a muscle at rest must be what?
Negative.
Negative 70. Because what's going to happen then, when acetylcholine attaches, sodium ions? Good. It rushes in. What's that going to make the inside of the muscle?
More positive.
Positive. What do we call that when it goes from negative to positive?
Negative.
What?
Depolarization.
Depolarization. It's called. So you're telling me the muscle gets depolarized just like a neuron, right? So far, is this any different than nerve transmission? No, it's the same. It's just happening in the muscle now. It's going to take a little left turn now as far as what happens next. Let's go to the next one. It's getting good. Okay, don't click the Jensen one, click the bottom one. Oh, I'm so excited. Okay, you're going to do the step through. Whew, here we go. So this is neuromuscular junction. So this is a what? A synaptic knob, also known as
Axon
terminal. It connects to the muscle at the what? What's this?
Sarcolemma.
Sarcolemma, which is the cell membrane of the
muscle
fiber. Each of these are called myofibrils. What's from here to here?
Sarcomere.
Good. What's the yellow stuff?
Actin. Is it actin?
Sarcoplasmic reticulum, which it contains what?
Calcium.
Which has what charge?
+2.
Good. What's these tubes?
T2.
Good. Now, next, go. Yeah, the little fancy blinking one. There you go. Yep. All right, now press play. Now we're going to go through 10 steps. Just let it go. All right, so we're zooming in, and it looks like chocolate chip cookies. I'm aware. Action potential comes down, and it's going to cause what to come in? Calcium. It's going to attach to a vesicle. Cause the vesicle to fuse with? Press play. Nah, stop.
The S word.
All right, now it's going to zoom in. What it's going to show you is how vesicles actually attach to the cell membrane, and it uses something called snares. Go ahead and press it again. This is at a little deeper level, even than what I need to teach you, but they snare. They allow the vesicle to catch with the cell membrane. Go ahead. And you'll see the interaction here in a minute visualized. So let's play. Ooh, look at that. Action potential. What's coming in?
Calcium.
Calcium. What's it going to attach to?
Vesicles.
The vesicle. It attaches to something called synaptotagmin. I won't ask you that. If you know calcium attaches to the vesicle, smile, okay? So calcium attaches to the vesicle. What's inside of here?
Neurotransmitter.
Neurotransmitters called what?
Acetylcholine.
Acetylcholine. Abbreviated? ACH. Watch, go. Watch what happens. Calcium attaches, the snares interact, and now watch. It's magic. Woo! See that? What is that?
Diffusion.
No.
Darn.
What's called when it leaves the cell? Exo. Exo what? Exo what? Cytosis.
Darn.
Exocytosis. What got kicked out? Neurotransmitter called? Acetylcholine. Into the space called the what? Space!
Synaptic cleft.
Synaptic cleft. Acetylcholine attaches to?
The
Receptor proteins on the
Sarcolemma.
Your brains hurt yet? Yeah. All right, let's go. Now it gets good. Here we go. They open. What happens? Sodium rushes in and causes the entire muscle cell to become what?
Depolarized.
Depolarized. So it becomes what charge?
-7.
Positive
It
35.
becomes positive. Because what ion came in? Positive. Do you see the yellow charge go this way?
Yeah.
That means the depolarization travels like a wave all the way across the cell membrane of the muscle, across the sarcolemma. Eventually, it's going to run into
T cells.
the T tubules.
Tubules.
Let's watch what happens. Go. This is so good. Oh, but we can't leave it there for long, otherwise your muscle's going to stay contracted. Acetylcholinesterase. See those little Pac-Man looking dudes? What do they do to the acetylcholine?
Ate
It
it.
ate them. And it actually breaks them apart into component parts. And we need to get the acetylcholine out of there, otherwise what's going to happen to your muscle?
It's gonna cramp it up.
It'll stay contracted. Can't have that. So we got to get rid of the acetylcholine in the synapse. Next. There's another way. We also suck it back in by reuptake proteins, which is shown here. Watch. Go. Click it. Click it. This is good. Watch this. It comes in, and then it gathers. Go to the next.